Well, I'm Joe Guthrie. My co-author for this talk today is Angeline Meeks. I'm going to be talking about our project in the Predator-Prey program called the Corridor Observatory. Just a brief, okay, why are we slow advancing? I'm going to try not to do it twice. Is it the mouse?
Click on here with your mouse somewhere and now try it.
There we go. Okay. Thank you, Laura. Quickly, I just want to go over what I'm going to be talking about today. We're going to give a really brief overview of my involvement with the Florida Wildlife Corridor, which is some important context for what we're doing now with the Corridor Observatory. Be a little bit of a tiny bit of my personal history mixed in there, but the heart of the talk is going to be about the Corridor Observatory project. This is a map that was Josh showcased a bit earlier today in his talk, describes the current state of the Florida Wildlife Corridor with conserved areas in the dark green and the opportunity areas within the corridor in the olive green. These percentages on the right-hand side reflect the roughly 8 million acres of land that's yet to be conserved, along with about 9.7 million acres of existing conservation land that's already been put into permanent conservation. Rather than delve into the kind of long history of the Florida Wildlife Corridor, I thought I would encourage everyone to just go to YouTube and watch Dr. Swain's plenary speech at the 2022 Florida Wildlife Corridor Summit. It's far from being kind of dry history. It's really a fascinating case study in how these kinds of the kind of movement that we have now in Florida gets started and how it grows. If we had to point to a single person as the originator of the idea of a connected network of reserves in Florida, that person would be Reed Noss, who began publishing as a PhD student, began in Larry Harris's lab at University of Florida, began publishing the first maps of the vision back in the 1990s. The Florida Wildlife Corridor, as many of you know, is the product of decades of work involving thousands of people from the agencies, land trusts, private landowners, businesses. No one person or NGO can claim to have created the idea of the Florida Wildlife Corridor. It's something that we've all contributed to and given our energy and focus. This slide details just a tiny bit of my personal journey post-grad school in the early years of my career. The current corridor effort began to kind of change and become slightly more public facing around 2006, when our friend conservation photographer Carlton Ward began working at Archbold and began using his camera and his connections in two industries. Those was the cattle industry here in Florida and the publishing world. That's where he really began to document the unique rural lifestyle here in Florida, in our part of Florida. And it was kind of this work that Carlton was doing and the relationships he developed and maintained gave rise to a series of self-powered expeditions that's actually ongoing to this day, where the group of us kind of together set out to kind of highlight the story and highlight the real possibility of a real Florida Wildlife Corridor, protected Florida Wildlife Corridor. So in a sense, we kind of decided we would use our own bodies and our own energy to kind of measure the scope and scale of this massive thing we call the Florida Wildlife Corridor. We also leaned heavily on the documented movements of certain iconic wildlife species like the Florida black bear, which was the subject of a subject of a study here by the University of Kentucky between 2004 and 2012. And I'll say just that, if we bring it to present day, we now have a series of 5 films, 2 books, kind of innumerable news hits, news stories on our expeditions, kind of fueled by our expeditions, and we've kind of begun to build a mass of support for the Florida Wildlife Corridor. But much of that, as I mentioned, kind of much of the science that undergirds the corridor conservation effort was really informed by telemetry studies on those large, wide-ranging wildlife like panthers and bears. So now we're engaged in an effort to bring new science to the Florida Wildlife Corridor conservation effort, and we're calling this new science the Corridor Observatory. Our goal with this project is to conduct really collaborative science that engages the public and gives a model that other conservation organizations might use to scale up to efforts that are even larger than the Florida Wildlife Corridor, to perhaps national efforts. So we have an opportunity to, we have an opportunity here to collect data using the latest technologies to kind of further fill in our picture of the trends and relationships among species across the landscapes of the corridor. Talking about camera traps, talking about bioacoustic monitoring. One of our contraptions is pictured here, but we want to use kind of non-invasive methods to capture a broad suite of wildlife to tell us what's going on across a whole broad spectrum of landscapes within the corridor. So what are we talking about? Well, we've found 3 study sites to initially kind of roll out this corridor observatory effort. And those are Archbold Biological Station, the Archbold Complex, Buck Island Ranch, and the DeLuca Preserve, which is owned by UF, 26,000 acre reserve in Osceola County. This zoomed in look here kind of shows the intensity with which we're collecting these data on our 3 sites. Again, the Archbold Complex as you look at the map on the left, Reserve, McJunkin Tract, and Lake Placid Scrub. Each camera is co-located with an ARU, which is a autonomous recording unit. So our ARUs are produced by the Cornell Lab of Ornithology, and the cameras are Reconyx Hyperfire cellular cameras. So you can see we've got quite a number of them out in the field. There are 135 cameras we're managing and somewhere around 107 ARUs. We could not do this without a large team of people, and very important in this effort is the University of Florida's Range Cattle Research and Education Center, where Dr. Hans Ellington is assistant professor of wildlife in the Rangeland Wildlife Ecology program. Hans's team has helped us roll out the DeLuca Preserve network. We're also working very closely. I haven't given a shout out to our data and technology program here at Archbold, but we work very closely with Viv's team, and you'll understand a bit more why in just a second. In this graphic, kind of get an idea of the tremendous amount of data that we're collecting across these 3 sites. First, at Buck Island Ranch, it needs a little explanation. The Buck Island Ranch network of cameras has been in existence since 2015. This is the work of our colleague, Dr. Raoul Boughton, which is funded and operates through a cooperative agreement with USDA. So since the initiation of that project, we've amassed around 9.5 million images just on the camera side. Those cameras are now joined with, we placed 16 ARUs in the field with those, and they've begun collecting data for about the last 150 days. The DeLuca Preserve camera network is 48 cameras along with 48 ARUs. You can see the tremendous amount of data that we're getting. All those terabytes are coming from the sound data. We're operating these devices in this first year 24/7. So Matt and Viv here from the data and technology program can attest to just what a tremendous amount of data we're collecting. But we hope we will slowly kind of refine that data collection and hone it down to something a bit less data heavy as we figure out what are the times of day where we can get the most data and kind of hone that down. All of this data we intend to send to the cloud where it can be managed. The organization we're working with now is called Cyverse. We're very optimistic about this. Cyverse is an NSF-created open science workspace for collaborative, data-driven science. and it has all kinds of tools and resources for allowing data management of this type of data collection, huge data. So we're excited about that, and we're going to let everyone know how it goes. So far, I'm not at the point where we can present any, obviously not at the point where we can present any results. One of our early results is just to show that this can work. But we're excited to have, in the early going, collected documented evidence of 19 mammal species, somewhere around 34 bird species, including some more icons, the wood stork, the scrub jay, lots of stuff here. And so we're producing a ton of imagery, even some imagery on some of the reptile species. The poor striped crayfish was captured in probably one of his last moments of life there in the lower right. So we'll continue to try and marry the kind of heavy duty science with a consistent effort at storytelling. Our data are their data, make no mistake, but they also happen to be pretty engaging tools for our outside audiences, for followers on social media. So we're working closely with Archbold's communications, as well as UF communications department to use the images and sound from the corridor observatory. We like to say that we're providing the eyes and ears for the corridor to the public. We have a lot of next steps. We have a lot of work ahead of us, but now that our equipment is working in the field, we're very serious about developing the processes and protocols that are necessary for managing this much data. So our next steps are really to firmly establish our data pipeline, getting data off the instruments, transmitted over the cellular network, or retrieved by hand and uploaded to the cloud where it can be managed. We need to one of the early steps is we're going to need to develop training data sets on each species of wildlife and develop lists of our target species and focal species for this. That workflow, I was going to say, will need to involve a method for tagging the metadata to each image or each bit of sound. So we need the species ID, we need things like the habitat, we need location added in there. So there's a tremendous amount of work that we have to do to add all those data in. So quickly, I just want to acknowledge the already large and growing group of professionals who are helping us do this. I want to give a special shout out to Emily Angel in the Predator Prey program. Emily's really our great field marshal for the monthly effort to service all these cameras and keep everything running. Same goes for the UF team, which is managing everything at DeLuca. Finally, I want to give a special shout out to a really special person from the UF team, Beth White, who tragically passed away in 2022. Beth made huge contributions to this project and a great many other projects as a research biologist. She knew Archbold. A number of people here knew and worked with Beth, especially at Buck Island Ranch, which was one of her favorite places. Every time I saw Beth, she wanted to know what was going on at the ranch. So she was just a really wonderful colleague, and we miss her greatly. All right. Hopefully I got into the finish line, and I'll give it to any questions.
So, any quick questions from the room or any online?
Great.
Go ahead. Yeah. Go ahead, Amartya.
So you have all these terabytes of audio data on it.
Yeah.
So do you actually physically listen to them? Or do you have some kind of software which looks at?
So Marcus' question is, with the volume of data, are we actually listening to them? No, we have software that's provided by Cornell. There's another software called Raven Pro that allows us to kind of process the data with the software. But we do have to create those training sets. So human reviewers have to listen to a lot of data to identify enough so that they can create a model training set. And then that forms the basis of the algorithm that we process everything with.
Great question. So thanks again, Joe. And Our next speaker, that's right, it's online.
Okay, Jeremy, go ahead and start sharing your screen and I'm gonna get you onto our big screen here.
All right, that sounds good. And also you can hear me, right? Just want to make sure
Yes.
of that. Perfect.
Set to go, Jeremy. Introduce yourself and introduce your talk. Thank you.
Perfect. All right. Let me just get this. Nice. Okay. So thanks for having me here. I'm Jeremy Summers. I'm a graduate student in Nancy Chen's lab. And today I'm going to be
Jeremy,
presenting.
you might want to put it. Yeah, you got it now.
It was just delayed a minute. You're good.
Okay. Okay, cool. So everyone, as I was just saying, I'm a graduate student at Nancy Chen's lab, and today I'm going to be presenting the work that I've been doing on examining the factors that lead to population stability in the Florida scrub jays at Archbold, and what that might have to do also with the immigration rate of birds into the population. So the research that I do is primarily focused on the question of why populations persist in the first place, because it kind of makes sense that populations that are growing can continue growing exponentially. Whatever factors are causing that growth will feed into that growth. And the same can also be said for decline, where decline can easily feed into other factors that will lead to further decline in a population. But stability actually requires a combination of factors both pushing the population to grow and decline. And there is both density independent and density dependent factors required for this. And for density independent factors, there's a requirement for them to cycle between different values in order to create that trend of constant stability. So climate has to be good one year, bad the other, for the population to remain stable over time. Density dependent factors are self-regulating. So if there's like a density dependence factor, then if the population grows too much, it will then shrink the following year. But then there's also factors that kind of bridge between these two groupings of density independent and density dependent, such as food availability that can respond to the population growth, or also individuals moving between populations. And I'm primarily interested in this connectivity between populations. And this partly has to do with the fact that populations becoming isolated, that connectivity being cut is a major issue for species across the globe. The amount of land cover change across the world has been increasing dramatically over time, and many species that were previously thought to be stable populations can actually undergo decline when immigration is cut. This is a cute little example that I have of a woodchat shrike population of Germany. Had a long history of being stable, but as soon as immigration was cut due to land cover change in the area, that stable population actually went to extinction. So I'm really curious then about how to explore the effect of changes in connectivity between populations in this broader context of what creates population stability. And the Florida scrub jays are a great system to be investigating this. They, of course, are highly philopatric, so there's plenty of data monitoring the population, and the population at Archbold has a huge plethora of data that I can use to investigate the demographic history and what factors might be changing that over time. It's also an extremely stable and well-preserved population, but the surrounding populations are rapidly declining, presumably, as the immigration rate has also rapidly declined over the past 20 years. The number of new immigrants dramatically decreases over time. And so then the question would be, is this population remaining stable? And if so, how is it remaining stable as it has become more and more isolated from surrounding populations? To investigate this, I created a model to model the demography of the Florida scrub jay that is affected by many different factors. So starting out just looking at the population growth rate of the Florida scrub jay over time, with one, this blue line that I have on here, being that the population has remained stable over time, the actual population growth rate over the past 30 years has been roughly at 1. It's been very, very stable. But from each individual year, the population growth rate has rapidly fluctuated between less than 1 decline and more than 1 growth. So the population stability is created by rapid fluctuations in the population growth rate, which is pretty common in a lot of species. And so I was really curious to be able to investigate what is creating these fluctuations in the population growth rate over time. And to investigate that, I used a matrix population model. These models take different vital rates about the population, such as survival, fecundity, and maturation, and defines how that allows individuals to move between different biologically relevant stages within the population, like how an individual survives to become a juvenile, to becoming a 1-year-old individual, and then maybe even a breeder if they're able to mature into that stage. And I also included immigration into this model. And immigrants can enter into the population either as non-breeders or as breeders, because the Florida scrub jays have their cooperative breeding system in which there can be non-breeding adults within the population as well. I then took my matrix population model and I used a life table response experiment to investigate how the variation in those rates, not just the rates themselves, influence the variation in the population growth rate. Because you could have a rate in the population that is extremely impactful, but if it never varies, it's not actually going to result in the population changes that we see over time. So a life table response experiment takes the variation of the population growth rate and breaks it down in terms of the variation in each of those demographic rates, scaled by the effect of those demographic rates on the growth rate. But I can also go a little bit further because the plethora of data available at Archbold. I can use the environmental data with generalized linear mixed effect models to investigate how the variation in those environmental variables affects demographic rates and then affects population growth. So the variables I was able to include were things like acorn abundance to measure resource availability for the population, the area burned as a metric for habitat quality. I used the total area burned within 5 years for this. The population density, this was just the number of adults per hectare. The equatorial southern isolation index, which is just a measure of the El Nino La Nina cycle, which includes factors such as precipitation and temperature. So this is sort of my climate factor. And then also the year, to account for any temporal trends in vital rates. Most importantly, that includes immigration. We know that immigration has a negative trend over time. So that would be captured by this year term. And I could break down the variation that I measured for the population growth rate into sort of 3 different factors of variation. Some of that variation is explained by variation in those environmental factors acting through the vital rates. And some of it will be explained by not those environmental factors. This can include density dependence that isn't captured by the number of adults or other interactions between individuals of the species or other environmental variables that weren't modeled in the model. But then I also included effects of past years. So this is a measure of how demographic structure, so how the population is doing a previous year, affects the growth this coming year. So those are the 3 different ways in which variation can be measured using my model. And actually presenting the results, this large orange bar is showing the amount of variation in the population growth rate that is explained by fecundity, variation in fecundity, broken down by that dark bar, the environment, the slightly less dark bar, which is the variation within the focal year, and the lightest, that demographic structure bar. So variation in fecundity makes up almost 60% of the total variation in the population growth rate. So fecundity is really what's driving those rapid fluctuations in the growth rate that we see. But this is then followed up by survival. But survival also has a much larger proportion of its variation made up by that environmental variance. And the variable that I was most excited to sort of investigate is immigration, which has a very, very small amount of an effect on the actual variation in population growth rate, with absolutely almost no effect from the environment, which includes that temporal trend that I want to investigate. So let's go a little further and actually see how the different individual environmental variables create those darker bars that we see. So sort of each of the bars that I presented here in different colors is sort of a zoom in of the darker bar from the previous plot. It's broken down by the different environmental variables that I included in my model, and the negative portions on it are cases in which covariation between 2 different vital rates affected by the same environmental variable actually reduces variation in the population growth rate. There's a very large effect of EQSOI, so climate plays a large factor, especially in fecundity and survival. And there's a large effect of acorn abundance, and that's actually primarily through juvenile survival. But when I actually add in year terms, temporal trends almost cancel each other out entirely, especially for immigration, which explains why there's so little effect of environmental variance through immigration. So what if I then zoom in more, because I said that there was covariation between vital rates on the bar for immigration. When zooming even further, so this is covariation between immigration and other vital rates that are both being affected by the same environmental variables, we get this large variation from the year. So that's that negative trend in immigration. But then there's also variation in other vital rates over time. This negative trend over here that we see in EQSY means there's a negative trend over time in EQSY over the time period that I looked at, that co-varies with the negative trend in immigration. So this would be like there's covariation between year and EQSOI, which both have effects on immigration and survival, which in turn both affect population growth in opposite directions. And then there's also the negative effects on the different vital rates, which indicates there's opposite trends in fecundity, survival, and transition rates that are counteracting the decline in immigration. So taken all together, the results that I found show that fecundity is what's really driving these fluctuations in the population growth rate over this long period of time, and that the environmental factors do play a major role in this population, and they primarily affect survival and to a large degree. But then this aspect of immigration is that this decline in immigration appears to be coinciding with other trends and other vital rates, which are allowing the population to remain stable over the time period investigated. I'm going to be expanding this analysis to further years in the coming future and seeing if any of these trends might potentially be coincident with other vital rates. But it would seem to indicate that this well-preserved population is being able to buffer itself against isolation from a decline in immigration. And that I would be happy to take any sort of questions. And I would like to thank everyone that's allowed me to continue on this project. Everyone in the 10 Lab, everyone at Archbold has also collaborated and has provided the amazing amount of data that is available through this. I'd be happy to take any sort of questions.
Well, thanks for that talk. And are there any questions from the floor? Well, maybe I could start with one. So you looked at the Southern Oscillation Index, although it might be worth also looking at the other indices that drive things at Archbold, which is the AMO, which you probably know, and the PDO. But actually what I'm really interested in is how do they interact with other factors such as fire and acorn production? So it always seems that these indices are much better indicators than, you know, individual things like rainfall. What's your thinking on that?
Yeah, so I did a large investigation of different indices, seeing how they were correlated with each other and what environmental variables they also correlated with to minimize any use of overly correlated variables within my model. With the relationship between other environmental variables and the Southern Isolation Index, interestingly, I didn't find any sort of relationship with area burned, but the previous year's Southern Oscillation Index is still heavily correlated with the acorn abundance, which makes sense. Acorn abundance within this is being measured within the fall, and the years that I was using within this model are starting at the breeding season. So Southern Oscillation Index does seem to relate to directly to it, those wetter years providing a larger boom in acorn abundance. And both of those have this alternating pattern, which I said at the beginning helps them to promote population stability over time.
Very interesting. So any other questions on that front? Well, if any from the chat, no. So if so, I'd like to thank you very much, Jeremy. I suspect you'll get some follows up from avian ecology as well, and we'll move on to the next question. So thank you.
Okay, hi everybody. My name is Betsey Boughton. I'm the program director of Agroecology. Thanks everybody for sticking in there all day. I know it's been a lot of information. So today I wanted to give an update on the long-term agroecosystem research network, because you might have heard about us talking about it, and you might hear LTAR. And I thought, well, maybe not everyone knows exactly what this network is about. So I thought it was A lot of our staff work on LTAR, so Viv and Shefali and a lot of people at the ranch are involved in various ways. And of course, Hillary is our site lead, and we work with our partners at University of Florida as well. So it's a really big effort that we're all working on together. So the long-term agroecosystem research network was started in 2012, and it really came out of this need for coordinated agroecosystem research. Agriculture covers about 38% of the world's land area, and over 50% of the US is in agriculture. So this such a large land use combined with our growing population creates this need for agriculture that provides more than just food. So increasingly we're thinking about land sharing, where we might be producing food at the same time as trying to achieve some conservation goals. And so that's what LTAR, one of the goals of LTAR was to understand how we can increase production of food at the same time as enhancing soil, water, air quality, biodiversity, habitat, and ecosystem health as a whole, and making agriculture as economically viable as it can be and socially responsible. So given that across the US, we have a lot of different diverse types of agroecosystems, and so LTAR was initiated to assess all of the diverse agroecosystems and what it would take to achieve these goals broadly across the US. And we also work with networks in Canada and Europe. So this is also starting to be a global effort. So the LTAR overall goal is to develop national strategies for sustainable and resilient agriculture. And the network currently comprises 18 sites. You can see them in the map here. The brown sites are grazing land. The blue are integrated systems with both crops and livestock. And the green are cropland areas. And they represent a large part of our agriculture production throughout the US. Co-production with stakeholders, as Aaliyah talked about earlier today, is really integral to the LTAR's approach. And the idea is that if our partners are involved, especially producers, and if they're engaged in the co-creation of research priorities and involved throughout the whole process, then our research innovations that are identified will more likely be adopted. So this network is really big, and we have over 700 people that are involved in some way or another in the research of LTAR. And so how LTAR has dealt with this huge interdisciplinary working group interdisciplinary research is through working groups. And so here I've listed some in green, and we have over 28 working groups in LTAR. So like Aaliyah is in the human dimensions group, Amartya is involved in the water quantity and quality group. We have Aaliyah and Shefali involved in human dimensions, and we have Viv involved in remote sensing and GIS, just as a few examples. I'm co-leading the biology group and also the grazing lands working group. And then these are some of the research products that have come out of LTAR. So what we're trying to do is gather data from our diverse sites across the US and look for general patterns and understand what's happening in these agroecosystems across these broad climatic gradients. So these cross-site working groups allow new ideas to emerge from multiple sites, and then they're shared across the network. So there's a lot of information sharing that occurs across this network that is beneficial to all of the sites individually. And then a big initiative within LTAR is the Common Experiment, and it's called Common because all the sites are tasked to implement the Common Experiment. And these are 18, now 19, because we have a site in California, but it's not on the map yet. These are 18 regionally specific comparisons of different strategies. And what the core of the experiment is to compare an aspirational production system versus a business as usual. And within this experiment, our goal is to assess the trade-offs among ecosystem services. And some examples of aspirational practices include cover crops, no-till, crop diversification, integrating of grazing into crops, rotational grazing, prescribed fire, adaptive management planning, rangeland restoration. So there's a lot of diverse aspirations going on across the network, and it really depends on what region you're in for these regionally appropriate treatments. So one of the key functions that makes the network function is the use of these rigorous data sets where you can compare and put into big analyses and run analyses at the network scale. And LTAR has leveraged common sensors and protocols where possible. We've even adopted some protocols from NEON. However, because our research is so stakeholder driven and because our agroecosystems are so diverse, we recognize that common metrics are not always possible. So what we've been working on, this has been a huge effort, is the development of common indicators that represent sustainability, that we are looking at as a holistic measure of sustainability, but the actual metric to measure those indicators may vary across the network. So you can see that there's a whole host of these different categories that represent sustainability: environmental, economic, and social. And then within those 3 domains, we break it down to atmospheric health, land and water health, production and abundance. That's like the yield, the food provisioning, the financial stability. And then there's a lot of social factors that go into sustainability. These include human health and social cohesion of communities. This is a very complicated diagram, and it's taken a while to come up with how we can measure these and analyze them at the network scale, and it's still ongoing. I'll just give you one example. So the biodiversity indicator here, is you see those little 2 dots outside, those represent the metrics that they might be different across the sites. So a biodiversity could be a key indicator across the network, but it might be represented by different species or functional groups depending on the site. So it might be pollinators at one site, or it could be grassland birds at another. And it really depends on the type of agroecosystem and the stakeholders. And ultimately, these metrics and indicators will be used to compare business as usual and aspirational across the network. So I want to dive down into what we're doing at Archbold and University of Florida for our common experiment. And as Aaliyah mentioned earlier, we're working across the land use gradient of our grazing lands. So the lowest intensity management intensity of grazing lands in Florida is native rangeland. And then sort of a middle ground is semi-native that hasn't been fertilized or highly converted, but it has had some modifications like ditching. And then improved pastures, which are the highest management intensity. They're completely modified pasture systems. And what we're asking across our three land use types is how does an innovative management affect cow-calf production and multiple ecosystem services? And the management that we're focused on right now is fire. So at Buck Island Ranch, the aspirational or innovative management that we're testing is called patch burn grazing. And we're comparing it to business as usual, which is called full burn. And in patch burn grazing, instead of burning the entire pasture, we only burn a third of the pasture. And each year we'd burn a different third. And this has been tested in the Midwestern Great Plains as a way to improve grassland habitat and increase biodiversity. And it's because when you burn a third of the pasture, it is like a magnet drawing cattle to graze on that area. And they sort of leave the other 2/3 of the pasture alone. So you get the creation of short grass habitats and tall grass habitats. So you basically improve heterogeneity in your pasture compared to a full burn system where the entire pasture is burned. And we're testing this at Buck Island Ranch. We have 16 pastures. 8 of them are in the improved pastures, which are shown in green, and 8 in the orange, which is the semi-native pastures. And we have 4 replicates of each treatment. Measurements we're making include a whole host of vegetation measurements where we're looking at plant diversity, the biomass, the forage nutritive value, soils, which include a lot of soil nutrients and soil carbon, cattle responses. We monitor how cattle behave in these 2 different treatments and also their body condition. We're also really interested in greenhouse gases and measuring carbon, methane, and nitrous oxide flux, and water quality. We monitor with lysimeters, which are a way to sample leachate, and we also periodically measure birds. Okay. So some of the results from this project so far, we have published one paper on how patch burn grazing impacts forage, and in this graph over here, you can see that the red line is the recently burned patch, and they have actually higher crude protein and digestibility compared to unburned areas. But what we found was that patch burn grazing is much more effective in semi-native pastures compared to improved pastures. And I'm happy to discuss why that is if anyone's interested. At the UF Common Experiment, they're working in the native rangelands. So these are pine flatwoods. And the aspirational treatment is flatwoods burned every 2 years. And that's in green on the map. They also have 16 pastures. The business as usual is burned every 4 years. And that is in the red. The yellow is unburned, and the blue is a 4-year burn interval followed with a chopping after every burn event. They're also doing similar metrics as we are, so you can tell this is a really holistic measurement of the ecosystem. We're measuring lots of stuff, and we really our hypothesis is that the aspirational is going to be more provide more ecosystem services than the business as usual. And then here's just a quick result from one of Maria's work in the native rangelands. And you can see on the x-axis, we've divided into different functional groups that ranchers are interested in, and then the biomass on the y-axis. And saw palmetto, for example, is much higher in unburned areas and does decline with burning and chopping. And then you can see how these other grass groups or vegetation groups vary among the treatments. Lastly, I just wanted to mention our strategic plan. We have just developed this in 2022, where these are the research initiatives in the blue. We have a big focus on science for climate smart agriculture. We really need science-based information. There's already being money invested in climate smart beef, for example. But LTAR is well poised to develop the data and the analysis to actually test if some of these practices do make a difference for the climate. And I'll just leave it at that.
Well, I'm sorry, Betsy, we don't have time for questions, but please follow up with Betsy or put some questions online. Our next speaker is a guest who's come up from Miami, who's going to change directions a little bit for us. Has been an artist here on and off for multiple years. So Deborah, over to you. Introduce yourself briefly and what you'd like to talk about when Deborah's got it right.
How's everybody doing? Okay. Do you need to stretch a little bit? You can stand up. I'll take 15 seconds off my talk. So get it. I think we only have one more after this. It's been an amazing day. Here we are. That's me. Yeah.
All right, let's get.
So as Hillary said, my name is Deborah Mitchell. I've had the honor of coming to Archbold for about 6 or 7 years now. But of course, because of the pandemic, I've only been here once in the last few years. So coming up for a couple of days to share some of my work with you is truly an honor. So thank you for having me. We're going to do things a little different than we've done in the past. Hillary, usually she'll say to me, let's get your science-based work and share that. And we've had some success with Zoom over the last couple of years doing that with my various exhibitions. But today I wanted to really maybe share some ideas with you about how you might get your research out farther and wide to people. Whether you are just beginning your scientific career or perhaps you're embarking upon retirement and happen to be an amazing artist like we all know you do have here, I think we're going to have some good tips here. So I was born in Canada. I live in Miami Beach with my family. I recently became a certified master naturalist. That's how I spent my time during the pandemic, which means, by the way, that I really don't know much, because every time I come to Archbold, I feel just overwhelmed with the amount of information that you have here. My creative process is based on experience-based field work. So I go out in the field, that informs my practice, and go back to the studio where those observations are turned into actual work. My mediums include photography, painting, installation-based work, and also some drawing. Good. So I want to give you some framework, some context for what we're going to see in the next 13 slides or so by looking at the last couple of exhibitions that really had very, very large attendance numbers, as you can see up there. This was the first in a series that we did at Miami International Airport, and it's a good example of how work at Archbold fueled my creative process. It's called Wild Observations in American Flyways, and Hillary was a wonderful mentor for me on this. It combines scientific research with artistic interpretation, and many of the 42 pieces in this series are based on the trips here. For example, a few years ago when I was attending this actual symposium, there were 2 presentations on owls, and I was smitten, charmed. In addition, I would go back to my cottage at night, and there was a great horned owl perched out there, calling all night long. No one knows why. It just wouldn't stop. So I became rather enamored with that. I was able to go ahead and talk to Reed about his research, photograph some owls, owl wings in your specimen collection here. And that became, let me see if I can, that became this little piece right here. So, and this piece right here, tribal invasives, you'll see later in the presentation, was based on how ambrosia beetles are contributing to the problem of laurel wilt. So I was able to go into Mark Darab's lab and talk to him about all the wonderful insects he's been researching for years. So those are just 2 examples of 2 great people that you've had here on staff that have influenced my work. One of the things that I hope you'll see in some of the images is I definitely intend to find a lot of joy and have a lot of fun when I'm in the field. I can go out and get lost and just stare at a leaf for a while, that's true. But make no mistake, I'm passionate about it and want to find joy. And I think I've tried to do that over the years very much. This show was almost like a retrospective. It was in Everglades National Park for 6 months, site-specific locations, and essentially it's an expanded version of that airport show that you just saw. And in the next slide, you'll see how humans and animals interacted with some of the work that was outdoors. All of the work had QR codes that one could scan if they wanted more information that would then connect to scientific support of the work. You can see here Hillary up there in the top with Evelyn Geiser from UF. We've got Cornelius Tulloch is now an employee of ARI, the Artist in Residence program in Everglades National Park. That's a fantastic program. I was a director for many years there. It's a great example of how we could bring Hillary, I think over 27 artists we brought up here over the years, thus influencing their practice and taking the science back to their various jobs teaching or in gallery exhibitions. So that was really a wonderful tool and way to get the research out. Homeschool children were able to come to the field and see and explore the work that, yes, had been eaten by the vultures. The vultures went nuts on everything. It was most unexpected, but kind of fun, actually. We had Audubon folks from Key West here camping overnight, and we did tours with them. Friends of the Everglades, so your general nonprofit groups would all come down and see the show, and I could go down almost every week over 6 months and tour with everybody. It was really, really quite phenomenal, as well as members of the local indigenous community, a little bit of scat on some of the work. I don't know what that was. We can debate that later. And one of my favorite groups right there in the center came to see the work. It's called Leap for Ladies. So this is a program that takes previously incarcerated women and helps them reinsert into society. And one of the things we were doing was getting them out into nature and just being with these ladies who hadn't maybe seen alligators before, and taking them through the work was one of the most rewarding parts of my year, hands down. So it was quite a wonderful situation. So part of the show was also shown in Key West, where a group called 21 Sea Museum Hotels. Has anybody heard of that? Yeah, so Joe, it's quite wonderful because this group in Louisville, Kentucky, has gone over about 4 or 5 states in the South, and they take old urban cores that have been abandoned, buy wonderful buildings, make them boutique hotels, and then put contemporary art in them. So that's where about 11 pieces from Wild Observations went that were based from Archbold. So it's not like they're in somebody's home. They continue to inform the public. This particular slide that you're looking at is an example of a collaboration with a wonderful choreographer from National Water Dance. On Earth Day, the dance was live streamed across the nation. And for the first time, they included visual art. So I collaborated with a seminal artist named Wilson Bowers, who is doing amazing things, even in Tallahassee, massive murals. It was just fabulous. And one of my largest pieces to date was, I guess, about a 45-foot textile piece that came down there. Talk about joy. One of the highlights of that particular day was at the end of the choreographed dance, the piece, everybody was invited to dance with the professional troops. And there was a lot of emphasis on disabilities. So we had wheelchair dancers in the middle of the park. It was something I'll just never forget. It was a perfect example of kind of reaching outside of your borders and collaborating with people that you didn't think necessarily would work for your particular specialty. On that note, one of Hillary's recommendations was that I visit Rocky Mountain Biological Lab out in Colorado once. And when I was there, they did a call for me to see if I could shadow any of their scientists in the field. And Ben Blonder, who's now at Berkeley, I wanted to shout out to him because he does a wonderful blog. So that's a great way to get his climate change-based plant ecology work out to artists and even the general public. So he's somebody you might want to check out too. So another thing you have to remember when you're thinking about different ways to get your work out is urban corridors. So last winter, I was able to do this installation. It's called the Vitrine installation in a historic building in downtown in the heart of Miami. And I was able to focus on this imaginary dialogue with John James Audubon. And it was really fun to work on. You'll see this piece right here is called my Endangered Species Cabinet. And I literally painted most of it one afternoon when I was listening to a Zoom presentation by your John Fitzpatrick here. So sometimes those Zoom things work out in mysterious ways. And for me, it really, really had on that one. And you'll see in some of these shows too, that the work spills over. A curator will visit and say, hey, Deb, can we take half of that show up to another one? Pahaoki in Fort Lauderdale, another urban center. So it's just great to keep connecting the public with what you're doing, and artists can be a really good tool for that. Right here, again, this is that tribal invasive piece that I mentioned. Mark really helped me on to understand more. It was selected by the Kennedy Center for the Arts Across America series. This is a documentary produced by the Kennedy Center featuring Everglades Songbook Suite. It's a fabulous, iconic indigenous musician named Jose Elias, and he brought about 10 musicians out to engage us with a lot of call and response songs on Earth Day. Earth Day was very busy last year. And it was just very special. We had 10 of the slides. When I say site specific, we had like tiles that we put on concrete at trailheads in the park. So the art in this case really provided context for the audience as they were coming to have fun and enjoy the concert. So that will be shown, I guess, live streamed or streamed rather, on a platform this year. And I'll let Archbold know when that comes out, since so many of those pieces were really inspired by my trip here. This was just a show last month in Art Basel in Miami. Is quite a big to-do if you haven't heard from it. I put an installation of birds in a room. What we have over here is a new painting, kind of a large one, 6 feet, on grasshopper sparrow. I do wish I would have put a detail in there for you, because that little sparrow was right in there. Next time. This is a Cape Sable seaside sparrow that actually sold during the exhibition. And this one has kind of a cool story in that it is a pelican skull found near Arnsnicker Island in Florida Bay, north of the Keys there. And it had its tag. So we were able to trace its origin. And it started up in North Carolina. It lived to be about 4 years old, I believe, before it passed on this island where my friends found it. But I had created this bird dwelling at a residency in North Carolina, also a recommendation from Hillary. It was called Art Science in the Field, but I believe it's no longer operating. Anyway, they helped me find some reclaimed wood, and I made the bird dwelling and showed it in a couple of shows. So it really, really worked for that exhibition in particular. I'm almost at the end. This is where I want to highly recommend maybe a little bit of mistakes and failure. Reaching out of your comfort zone and doing something that maybe is just not feeling quite like you. In my case, I've been participating in these Zoom salon discussions with a bunch of brilliant people since September, although we're on our holiday right now. We resume in a couple of weeks. We're all really thinking about the Gulf of Mexico, the sustainability issues, and what we as a collective can do. So it's artists, it's scientists, there's even an astronaut there who's written a book. It's really quite something, a small cohort from Harvard too. So it's pushing me beyond my comfort zone. I've been drawing night and day, not just to try to interpret what we're discussing in the calls, but also because from the Art Basel show, they asked for a bunch of new work that had to be delivered last week. So I drew for 4 weeks straight, and it was actually kind of fun. Haven't drawn that much in a long time, so I'm really enjoying the process. So in my mind, this new work will be anchored by large-scale landscapes or waterscapes that I find dynamic and maybe beautiful or powerful. They move me. So I hope it will draw in the general public when they're exhibited. This one's already on its way to Switzerland. And then these pieces will be backed up or supported by smaller factors that are vital and necessary to the ecosystem. Sometimes it's just kind of fun. We all know how important oysters are, how they're filtering so much water all the time. But I got to tell you, I went fishing with a bunch of friends a couple of months ago, and I was catching dinner. It was a good day, but my husband kept bringing up just bunches of oysters. So sometimes it's just life getting in there and sneaking into the experience. And again, circling back to that idea that you've got to find joy in your work. Staghorn coral on the left, again, just a classic sargassum seaweed ball with a seahorse stuck in there, because I saw that day as well too. So actually got an email today, very exciting. I hope we're in the funding stages for this residency, which would be an exchange between myself and an Italian. And what we would do next year is I will invite this Italian artist over here. bring him to Archbold, take him around all of my places, introduce him to the people I know, and then I go over there and he does the same thing. In the end, it's a compare and contrast between coastal wetlands. Obviously, in the case of the Venice Lagoon, you're looking at a little bit more saltwater marsh systems. And then again, I take that work to Art Basel in Switzerland the month following. So that's where I am. What I wanted to do is just, again, not so much drill down on the science with the work I showed you today, but show you how if you reach out of your own ecosystem, your own network, and talk to people, make those connections, you probably will find some great entryways to share your research on large, unusual platforms. That's it. Thank you.
I don't think we've got, I'm so sorry. We've got,
I'll be around if you have
yes.
questions.
And I'd just like to shout out to Deborah for coming up today. We really appreciate it, and for opening our minds, both right and left sides, to the possibilities. So we've got one last speaker, Erin. Sorry, you're there. Well, I know you'll get us over the finish line well. So someone else. Well, it's always good at the end of a marathon. Yeah.
Great. Well, thanks everyone for sticking it out through the whole day. I'm excited to talk a little bit about some work that I actually started as a postdoc at University of Miami. So my talk is called Uncovering the Hidden Soil Fungi in an Ancient Endemic Ecosystem. I presented this at a couple of conferences over the summer. I'll start with some acknowledgements for co-authors, Damian Hernandez, who's about to defend his PhD, Eric Mangus, Viv, and then my former advisors, Chris Searcy and Michelle F. Kami. And then especially the MSA, Mycological Society of America, for funding a lot of this work. Okay, so in recent decades, there's been a real focus on microbes as these ubiquitous hidden drivers, which has been captured in various art forms, like this famous cover of Nature from about a decade ago, and also with plants as well, where you see these macroorganisms as not just the organisms, but just made up of all these tiny microbes that are inhabiting all the cells and interacting on the outside and in the soil and basically everywhere. And these microbes have enormous effects on the macroecology of the hosts, as well as influencing each other. And so a big question in ecology is just what are the drivers of many of these microbial communities? And in the scrub here at Archbold, those communities are really our endemic ecosystems, which are in large part structured by this elevational stress gradient. So in many of our white sands, we have this replicated gradient going from flatwoods to scrubby flatwoods to rosemary scrub. And we know these as distinct habitats, but trying to understand the microbes that are underlying them, which could be influencing the plants that are growing on them, is really important. And so I call this an elevational stress gradient. As you go up in elevation, you tend to have decreases in water availability, in nutrients, vegetation density, and fire frequency. And so the key question here is, what do the microbial communities underlying this very heterogeneous habitat of ours look like? And these include things like mushrooms. So the specific questions I'll talk about today. First, does habitat heterogeneity promote fungal and bacterial diversity? Second, what are the relative roles of environmental, spatial, and biotic drivers on the fungal communities in particular, but also a little bit with the bacterial communities? Next, I'll focus on fungal guilds, which I'll explain that a little bit later, and looking at how they differ across habitats. And finally, I'll talk a little bit on one of our former projects on networks and looking at how the stress gradient influences network properties. Okay, so what we did was we sampled all over Archbold Biological Station back in 2017. So we had focal rosemary bulbs where we would sample from, and then from there we would have triplicates of scrubby flatwoods and flatwoods. And at each of these sites, we would take soil cores, focusing on what we are calling the crust, which is the first 2.5 centimeters at the surface, and then the subterranean, which is roughly a foot down into the soil, to look at microbial communities down below. And from there, we took that soil, sequenced the DNA, so fungal ITS1 and the prokaryotic or bacterial 16S using Illumina-MiSeq. And so what we have from this is really great community data at 2 different soil depths, replicated at these 3 different habitat types all over the station. We sampled from 71 of them, and 40 of these groups ended up in our analysis. So the first thing I want to show is just that these habitats and depths have unique microbial communities. This is true of the prokaryotes and the fungi. So what you're seeing is an ordination of the community data for both of these data sets. The colors represent the different habitat types, and the symbols, the different depths, the crust or the subterranean. What you just take note of is that there is some nice separation of the colors and the shapes. So this means that we have these unique communities that are found across these habitats. And then the big question is, well, why is this? And so the next set of analyses tries to get at that. And so we were looking at these environmental, spatial, and biotic drivers. And for this, we use distance-based redundancy analysis, which is an ordination technique that allows you to also build in some hypotheses using covariates and categorical variables. So in this analysis, we used our soil depth, which I described already. We use 2 elements of the environment. So the elevation, which is using what we call this relative elevation, which is a proxy for the distance of the water table, and then time since fire. We also looked at spatial data using what are called PCNMs to actually use the distance from one another and actually test how that affects the different communities. And finally, we used data on the biotic communities, looking at the shrub community using a separate set of surveys that happened right where we sampled. Okay, so overall, when we looked at all of our soil samples, maybe not too surprisingly, we found that soil depth is the primary driver that's separating out these different communities. This is true for both the prokaryotes and the fungi. This shouldn't be too shocking considering the ordination plot we just saw. We could see that there is a real divide between the 2 different soil depths. So the next set of analyses, I divided up into soil depth. So first looking at the crust only, the fungi weren't actually affected by any of these significantly, so that's why they're not shown. But for the prokaryotes, interestingly, the biggest predictor was distance, or the best predictor was distance. So sites that were close together, regardless of their habitat type, actually just tended to have more similar communities than those that were far away. When we looked at the subterranean samples, these are the ones about a foot down in the ground, we see that the best predictor is actually the shrub community. And somewhat surprisingly, it's a better predictor than any of the environmental factors. So what this suggests is that that at this certain depth, microbes aren't really specializing to the environment so much as they're specializing to the plants in that environment, which I think is a very different way of thinking about microbial communities in the soil than we're normally used to thinking about. Next we looked at fungal guilds. So there's a handy software or a package out there where you can take your data sets and kind of run it through, and it can identify what we think are putative pathogens, various types of mycorrhizae and saprobes and that kind of thing. So I'm just going to show a few of the more interesting ones here. The first is looking at plant pathogens. So on the x-axis, we have our 3 different habitat types going up in elevation, and the proportion of reeds in the sequencing on the y-axis. And so what we see is that at the crust level, as we increase along this elevational gradient, we're seeing an increase in the number of plant pathogens, or at least the relative abundance of these plant pathogens, which is an interesting trend, especially considering that we think of this rosemary scrub as a very endemic ecosystem. It's a habitat that can it's by and large invasion proof, not entirely, but not many invasive species thrive in it. And so it kind of leads us to question whether maybe some of this could be plant pathogen related. So the next two are different types of mycorrhizae. You don't need to worry too much about the difference between arbuscular and ectomycorrhizae at this stage, but know that certain groups of plants will associate with arbusculars, particularly, well, most plants actually associate with arbuscular mycorrhizae. Probably the palmettos of all the shrubs are associating the most. And what we're seeing is, relatively speaking, in the flatwoods, we're seeing a lot of AMF. And that decreases quite substantially as you go upwards into the scrubby flatwoods and the rosemary scrub. And we see the reverse with the ectomycorrhizae. So these ones are associating with our oaks and pines. And you see as you move up the elevational gradient, we're seeing way more ectomycorrhizae. And so we're seeing this, this unexpected transition from arbuscular to ectomycorrhizal fungi. But when we actually look at our data, it makes a lot of sense. Where we find these ectomycorrhizae, we're also seeing oaks. And so that does check out. And so this is all sequence data. I'll just mention that Alon Tran, who some of you may remember, was an intern with us last year. She was leading a project to actually ground truth some of these findings. And so we were looking at 5 species of scrub oaks and hickory and using microscopy to actually look at the root colonization. So can't see that. This one. There it is. So this little black on the tips, these are colonized root tips. And actually, as it turns out, these oaks have a ton of colonized root tips, like basically all of them are colonized. But one interesting difference is the density of that colonization. So for the 3 oaks that were both in scrubby flatwoods and rosemary scrub, we see that as we move upwards in elevation, we see more investment in these ectomycorrhizal fungi associations. And this jives with what we saw in the sequence data. You move up this stress gradient, you get more of these ectomycorrhizae. Lastly, I'll just talk about a project using the same data set that was led by Damian Hernandez, who I mentioned is about to defend his PhD if he hasn't already, looking at networks. And so we were really interested in how stress relates to the stability of these networks. And so there's this idea that environmental stress can either stabilize or destabilize networks is really important, especially as we see all sorts of changes to the environment happening. And so this data set is a really good opportunity to look at that. And so the way Damian did this was by looking at two indicators of network stability. The first is modularity, which is the degree of compartmentalization into these groups of interacting or co-occurring species. So on the left, you see a highly modular network, and on the right, a much less modular. Ooh, spooky. A very spooky modular network. So high modularity is known to be a stabilizing force. So that's an indicator that we have stable communities if we have high modularity. The second indicator is called cohesion. This is really just the ratio of positive and negative associations. And more negative associations is known to stabilize the communities and prevent positive feedback loops. So when we crunched all the numbers, what we found is that the stress associated or was associated with decreases in these stability-related network properties. So both modularity on the left and this ratio of negative positive on the right. They're both negative trends, which is what we expect when stress is destabilizing the networks, which could have very important consequences as different environments get changed and we start to see potentially very destable microbial communities along with that. So just to wrap up, found that habitat heterogeneity across our habitats promotes diverse microbial communities. The biotic factors were more important than the environmental factors for predicting communities, particularly in these deep subterranean samples. We found this unexpected transition between arbuscular and ectomycorrhizal dominated communities, and the stress destabilized the microbial networks. This work is just an important step towards understanding the broader microbial landscape underlying the Florida scrub and understanding how it interacts with the plants that we find here. So thank you. I'll take any questions if we have time.
That's a great talk. Thank you. Any questions from the floor? I know We are nearly on time and this is Martia and then the one online. So Martia first, maybe Betsy, and then the one online. Come on up and just speak in. Betsy, maybe, who was the other one? You were.
I can repeat the questions too, as I say them.
So yeah, I was wondering do you know what might be driving the transition from arbuscular to active mycorrhizal?
I think it's a shrub related driver. So I think probably what's going on is that the shrubs are affected by the water availability and maybe some of the nutrient differences, maybe the soil type, and then the associations that they are able to form. So not all of them form these ectomycorrhizal relationships. I think that is driving then what microbial communities we find.
Very interesting.
Yeah.
There's an online question. Do you want to?
So this is a question from Dan who presented earlier. As someone else messing with the soil community data, not in the scrub, but over at the ranch, could you talk about why you chose ITS1 over ITS2?
No. I could not. That's a Damien question. I think they're kind of like, I don't know, I guess people get particular about them, but they're both used for these sorts of analyses.
What is
what it was.
ITS?
There are different regions of the ITS region, which are the more variable regions between the conserved regions, which is why they're good as markers for species ID.
Okay. So was there another one from that board? Well, thank you. Thank you very much, Aaron. That was great. So I'm going to bring this to a very rapid close, but I'd like to, first of all, thank all of you who are here who actually stuck through the whole day, and those who drifted in and out. Thank you very much. Secondly, I'd like to really shout out, there are still 23 people online. Could we give them some applause for hanging?